Hi everyone, today we are going to discuss about glycolysis. Glycolysis is nothing but splitting of glucose molecule into 2 moles of pyruvic acid. It is also called as EMP pathway because 3 scientists investigated the glycolysis. They are Emden, Meyerhoff and Paranas, hence called as EMP pathway. Glycolysis derived from Greek word glykes means sugar and lysis means splitting. Hence, splitting of sugar is termed as glycolysis. The overall reaction of glycolysis is this. Here, carbohydrate or glucose combines with 2 NAD plus 2 ADP plus 2 H3PO4 and gives rise to 2 moles of pyruvic acid plus 2 NADH plus H plus 2 ATP plus 2 H2O. The first step of glycolysis is phosphorylation. In the phosphorylation, glucose is converted into glucose 6-phosphate in the presence of enzyme hexokinase and Mg plus 2 ion. Here ATP is converted into ADP plus Pi, that is inorganic phosphate. This is phosphorylation. The second reaction in the glycolysis is isomerization. In this reaction, glucose 6-phosphate is converted into fructose 6-phosphate in the presence of enzyme phosphohexase isomerase and Mg plus 2 ion. Hence, it is called as isomerization because glucose and fructose have same molecular formula but they differ in their structures. This is isomerization. The third step involved in the glycolysis is phosphorylation. In this reaction, fructose 6-phosphate is converted into fructose 1,6 bisphosphate in the presence of enzyme phosphofructokinase. Here ATP is converted into ADP plus inorganic phosphate. Hence, this is termed as phosphorylation. The fourth step in the glycolysis is cleavage. It is the major step of the glycolysis. Fructose 1,6 bisphosphate, which is 6-carbon molecule, is split into 2 moles of 3-carbon molecules, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate in the presence of enzyme aldolase. Hence, the reaction is termed as cleavage. The fifth step in the glycolysis is isomerization. In the isomerization, dihydroxyacetone phosphate formed in the cleavage is converted into glyceraldehyde 3-phosphate in the presence of enzyme triose phosphate isomerase. It is called as isomerization. The sixth step in the glycolysis is oxidation. Glyceraldehyde 3-phosphate formed in the past reaction is converted into 1,3 bisphosphoglycerate in the presence of glyceraldehyde 3-phosphate dehydrogenase. Here 2 moles of NAD plus is converted into NADH plus H plus and 2 moles of 1,3 bisphosphoglycerate are formed. Hence, the reaction is termed as oxidation. The seventh step in the glycolysis is dephosphorylation. In this reaction, 1,3 bisphosphoglycerate is converted into 3-phosphoglycerate in the presence of enzyme phosphoglycerate kinase. Here, the ADP combines with the inorganic phosphate present in the 1,3 bisphosphoglycerate and forms ATP. Hence, the reaction is termed as dephosphorylation. 2 moles of 3-phosphoglycerate are formed in this reaction. The eighth step in the glycolysis is interconversion. 3-phosphoglycerate is converted into 2-phosphoglycerate. Here, the phosphate group present on the third carbon is changed its position to second carbon. Hence, the reaction is termed as the interconversion. It occurs in the presence of enzyme phosphoglycerate mutase. The ninth step in the glycolysis is dehydration. In this reaction, 2-phosphoglycerate converted into phosphoenol pyruvate by eliminating a molecule of water in the presence of enzyme enolase. Hence, the water is eliminated. This reaction is termed as dehydration. 2 moles of phosphoenol pyruvate are formed. The 10th step in the glycolysis is dephosphorylation. Here the phosphenol pyruvate formed in the dehydration converted into pyruvate or pyruvic acid in the presence of enzyme pyruvate kinase. Here the ATP is formed from the ADP and inorganic phosphate. Hence this is called as dephosphorylation. Hence this is the complete cycle of reaction, or it is the complete procedure for the glycolysis. In the glycolysis, if we observe, in the first and third reactions 2 ATPs are used, and 2 ATPs are formed in 7th reaction and 2 ATPs are formed in 10th reaction. So the 7th and 10th reaction is called as substrate level phosphorylation, and in the 6th reaction oxidation occurs. Here the NAD is converted into NADH plus H plus. This is the product or yield of the glycolysis. Thank you for watching. If you like the video, please subscribe.